Welcome everyone, and thank you for joining us today for our webinar titled Wireless Recording and Stimulation Technologies for In Vivo Electrophysiology in Conscious, Freely Behaving Rodents. This is Haley McCaffrey from Inside Scientific, and I'll be your host for today's event. Our session is sponsored by Triangle Biosystems International and will include 2 research case studies highlighting the use of new head-mounted implantable wireless sensors for investigating neuronal processes in vivo. First, we will be joined by both Dr. Melissa Caras and Dr. Dan Saines from the Center of Neural Science at New York University. Melissa is a postdoctoral fellow in Dr. Saines' laboratory studying how auditory perception changes as a function of experience, exploring the neural mechanisms that support its plasticity. Dr. Saines' laboratory studies the development and function of brain circuits to better understand how the development and plasticity of synaptic and membrane properties permit networks to properly encode auditory cues. Today, they will present a case study on auditory cortex recordings collected from freely moving gerbils during learning and task performance. They will share methodology, resulting discoveries, and discuss the importance of in vivo real-time comparisons of neural and behavioral measures. Following, we will hear from Bradley Barth, a PhD candidate in the Shen laboratory at Duke University. Bradley studies the neural control of motility in the gastrointestinal system with in vivo electrophysiology and electrical stimulation. He develops new devices and techniques to assess function and disease in the gut and designs predictive computational models and applies them to improve neuromodulation therapies for functional GI disorders. Today, Bradley will share a case study where they have achieved successful stimulation in the sacral nerve in conscious, freely moving, untethered mice using a fully implantable wireless nerve stimulation device from TBSI.
As was introduced earlier, we'll be talking about monitoring auditory cortical plasticity with a wireless recording system. So in our lab, one of the main questions that we seek to address is how auditory cortex neurons adjust their sensitivity to sound. And we ask this question in 2 main contexts that we'll be talking about today. One is perceptual learning, and the other is developmental hearing loss. So I'll be starting the talk off talking about perceptual learning. Perceptual learning is an improvement in our ability to detect or discriminate stimuli. It's important for a variety of complex behaviors, including speech and language acquisition. And some examples that you're probably familiar with from your day-to-day life include getting better with practice at being able to tell whether an instrument's in tune, or learning how to tell whether 2 sounds from a foreign language are the same or different from one another. So in the laboratory, there are many different percepts that we could use to study perceptual learning, but the one we'll be focusing on today is the perception of amplitude modulation depth, or AM depth. So all vocalizations, animal vocalizations, including human speech, consist of slow fluctuations in sound amplitude. And these fluctuations, these amplitude modulations, are important cues for speech comprehension. Now, in the laboratory, we vary the strength of those modulations, also known as the depth, from very strong, which is in this case 0 dB, to very shallow or weak, in this case -12 dB, all the way to unmodulated. And we do this so that we can assess an animal's threshold for amplitude modulation depth. So that's the smallest depth that's reliably detected by the animal. So just to give you an example of what these amplitude modulations sound like, I'll play 2 of them for you now. First, a shallow depth, -12 dB, and then I'll play you 0 dB, and you should be able to very easily tell the difference between the 2. So hopefully you could hear that the modulations were stronger and easier to detect in the 0 dB stimulus. Now, in humans, AM depth detection thresholds improve with training. So this is, again, perceptual learning. So what we're looking at here is an example of data taken from a paper by Fitzgerald and Wright in 2011. And on the left, we're looking at data from an individual subject. On the y-axis is the AM depth threshold, ranging from poor at the top of the axis to good at the bottom of the axis, as a function of training day. And you can see that with training, these thresholds for the subject improve. And at the group level on the right, you can see if you do a comparison between pre-training and after training, practice allows individuals to get better at detecting shallower and shallower depths. So we study AM depth detection and the perceptual learning of AM depth detection in the gerbil. That's our model organism. And the reason that we use the gerbil is that the range of frequencies that they hear overlaps quite nicely with that of the human. As you can see here, these are thresholds on the y-axis for various sound frequencies, which we perceive as pitch on the x-axis. You can see there's a nice overlap between gerbils and humans. This is unlike other commonly used small rodent species like rats or mice, which are shifted up towards much higher frequencies of hearing. So to assess AM depth detection in a gerbil, we use a behavioral task. So first, there's this broadband noise that's unmodulated, and that's playing in the background continuously. And animals are trained to drink continuously from a spout of water while in the presence of this unmodulated noise, which we'll call the safe stimulus because it's safe to drink. Eventually, the noise will transition to this amplitude modulated noise, which is one of the ones I played for you earlier, and this we call the warn stimulus. And when the warn stimulus is presented, if the animal continues to drink, they'll receive a mild aversive stimulus, a small electrical shock. It feels kind of like a static electric shock you get if you touch a doorknob in the winter. And they can avoid this shock if they simply withdraw from the spout when the warn stimulus is presented. So you can see on the far right here, when the warn is presented and the animal is off of the spout, they do not receive any punishment. This is a correct response. This is a hit. When the animals withdraw from the spout during a safe stimulus, that's considered a false alarm. And these two responses, false alarms and hits, are what we use to quantify the animal's behavioral performance. So we use a signal detection metric called D prime, and you can think of that as the difference between the hit rate and the false alarm rate in units of standard deviation. So just to give you a sense of what this is like, I'll play a short video for you now of an animal performing the task. This animal's already been trained on the task, so it's good at detecting the amplitude modulation. Okay, so that's the behavioral aspect in order to determine animals' perception of AM depth. To assess what's happening in auditory cortex, we use chronic electrode implants. So these are 16-channel electrode arrays arranged in this 4x4 grid from Neurometics, and the electrodes are affixed to a custom-built microdrive, which you can see on the left. So when the screw at the top is turned, it advances a cannula, a little piece of metal below the screw that in turn advances the electrodes deeper into auditory cortex after implantation. So this allows us to move the electrodes after the implantation and the surgery is complete. Now, auditory cortex in the gerbil is located pretty laterally on the brain. So in order to minimize damage to auditory cortex during the craniotomy and electrode insertion, we take a dorsomedial approach. So here you're looking at the top of a gerbil skull. At the top is the rostral end, or where the animal's nose would be, and at the bottom of the screen is the caudal end, where the animal's tail would be. And we're highlighting here lambda, which is a skull marker that we can use to identify stereotaxic coordinates for our electrode insertion. And our craniotomy site is typically located to the left, in the left auditory cortex over here. So here's an image from an actual surgery. By this point in the surgery, the skin and fascia have been removed and cleared from the top of the skull. Skull screws have been inserted in order to stabilize the final implant once everything's inserted. And a base layer of dental cement, that's that green stuff, has been put over the top of the skull, excluding lambda so that we can still use it for measurement purposes. And that dark purple square area that you see there, which is just me marking off where the craniotomy site will be. So this is after I've drilled out the craniotomy. The purple square in the middle floating there is the bone flap that's going to be removed. This is after the bone flap has been removed. You can see a nice healthy cortical surface. This is after I have positioned the electrodes where they're going to be implanted. You can see the electrode contacts, those circles on each shank of the electrode array. And this is after insertion. So after our experiment, we can do histology to confirm our electrode placement actually did hit auditory cortex. What you're looking at here is a rostral to caudal series of Nissl stain slices from gerbil auditory cortex, or gerbil cortex rather, with auditory cortex highlighted, and the red arrows highlight electrode tracks that entered auditory cortex. The dashed yellow circle in the final slice indicates an electrolytic lesion of one of my contact sites that I made after all of the experiments were finished. Okay, so during the actual experiments, we're using a wireless approach. So extracellular activity that's picked up by the electrodes are transmitted to the wireless TBSI head stage. And this head stage transmits the activity to wirelessly, again, to a TBSI receiver, which interfaces with a Tucker-Davis Technologies TB32. This is a channel digitizer. And from there, the signal is sent by a fiber optic link to Tucker-Davis, the RZ5 base station, where signals are filtered and processed in real time. So to quantify the neural activity, we use the same signal detection metric I spoke of earlier, neural D prime. And here, neural D prime you can think of as the difference between the firing rate evoked by the warned stimulus and the firing rate evoked by the safe stimulus, again in units of standard deviation. So here's a representative example of a psychometric function from one animal. On the y-axis, we have our behavioral detection metric, D prime. So higher D prime values indicate better performance. And you can see that the performance improves as the depth of the stimulus gets stronger, as it gets easier to detect. Here's a neural response recorded from an animal as the animal was performing the task. And you can see the amplitude envelopes of our sound stimuli in blue, and in black post-stimulus time histograms of this particular multi-unit. And you can see that the response to the AM stimulus gets stronger and stronger as the depth gets stronger and stronger. And we can convert these responses again into that neural D prime metric, and you see a very similarly shaped function where neural D prime improves as the AM depth gets stronger. And we can compare now single neurometric functions to individual psychometric functions taken from the same animal at the same time. And we quantify the threshold for both neural and psychometric performance as the AM depth that was required to achieve a D prime of 1. So that's those gray lines intersecting the neural and psychometric fits. So the question is, how do the psychometric and neurometric performances, how do they change as a function of training? So what we're looking at here is behavioral on the left axis, on the left plot, behavioral performance of an animal on the first day of training, and on the right, response from one multi-unit on the first day of training, recorded while the animal was performing the task. And you can see that both behavioral and neural performance gradually improve as training progresses. So we can quantify this in terms of, again, the AM detection thresholds. I've just schematized the behavioral thresholds here, but what you can see is that on the y-axis, again, our thresholds in blue are the engaged neural threshold. So this is while an animal is performing the task that I've laid out. And you can see that there's a tight correlation between engaged neural thresholds improving as a function of training day, and the magnitude and time course of this improvement matches what we've schematized here for behavior. What we're interested, though, in addition to just improvements in auditory cortical activity, is the role of a top-down mechanism in this process. So in addition to recording from animals while they are engaged in the task, as I've explained to you, I've also recorded from animals while they are disengaged from the task. So what I mean by that is immediately before or immediately after training sessions, I remove the spout from the cage and I play the same sound stimuli to the animals, but they are not performing any operant task. The electrodes are in the same position, however, so we're recording from the same cells, and again, we're recording responses to the exact same sound stimuli, just under this different context. So during engaged sessions, it's thought that top-down areas are actively modulating how auditory cortex responds to sound input coming from the bottom-up ascending pathway. During disengaged sessions, pre and post sessions, it's thought that these top-down inputs are offline. So the activity that we see in auditory cortex largely reflects just this bottom-up activity. So as an example, here's what a response looks like, again, during task engagement on the first day of training. You can see the stimulus amplitude envelope at the top in gray, a raster plot, and a PSTH plot of this one multi-unit. And during disengagement, you can see similar responses, but overall the responses are a bit weaker. The firing rates are a bit lower in pre and post sessions. And this reduced firing rate translates to a reduction in neurometric D prime values. So the neurons are less sensitive to amplitude modulation. Okay, so back to this plot that I showed you earlier. This is what I showed you happens for neural thresholds during engaged sessions. This is what happens during disengaged sessions. So you can see first that overall disengaged neural thresholds are poorer than engaged thresholds. And in addition, they don't improve to the same degree or at the same rate as engaged thresholds. And in fact, you can see that the difference between engaged and disengaged sensitivity grows progressively larger with training. And that suggests that during perceptual training, a top-down process that actively modulates auditory cortical activity is getting stronger.
Okay, so I'll take over now. A second form of long-term plasticity that we're interested in concerns changes to the brain and to behavior that occur as a function of developmental hearing loss. And here I'm going to first introduce the kind of behavior that we train animals to do. Our goal here is to assess both behavior and auditory cortex encoding of the stimuli following a long-term period of developmental hearing loss, and also following very good training of the animals. So we know we're recording from animals that are good performers. So the task here is a little bit different than the one that Melissa described to you. It's an amplitude modulation task, but here the animals are trained to discriminate between the rates of modulation. So all rates above 4 hertz are go stimuli, and all rates of a rate of 4 hertz is a no-go stimuli. It's an appetitive task, and you'll see the animal poke its nose to initiate a trial on the right, and then either go to obtain a water reward on the left if the stimulus is a go stimulus, or re-poke the nose poke if it's a no-go stimulus. So here are a few trials. So the animal goes to obtain a water reward because the stimulus was greater than 4 hertz. And the animal re-pokes in that case because the stimulus was a no-go. So once again, the overall task is to discriminate go from no-go stimuli. Go trials are those greater than a rate of 4 hertz. Animal pokes its nose. If it's a go trial, the animal can go get a water reward, and we characterize that as a hit. If the animal fails to get its water reward, that's a miss. On no-go trials, that is trials during which 4 hertz is presented, if the animal re-pokes and doesn't go over to the water spout, that's a correct reject. If the animal does go over to the water spout to try to obtain water, we characterize that as a false alarm. We turn the lights off. So just as with the AM detection task that Melissa described, we're using hits and false alarms to determine performance with the sensitivity metric D prime. The manipulation here is developmental hearing loss, and we actually remove a small middle ear bone called the malleus bilaterally just at the time when the animal's ear canals would open and it would begin to transduce airborne sound. So the animal that's about postnatal day 10 for most rodents, including gerbils. So the animal then has a permanent conductive hearing loss up until the time that we begin to train it and record from it. Conductive hearing loss refers to the fact that sound doesn't get into the cochlea, but as far as we've been able to characterize it, there is no damage to the cochlea or to the central nervous system. So any changes in the animal's behavioral performance or neural performance, we can attribute to changes within the central nervous system, not the cochlea. So this is the behavioral data plotted for the animals that we've run that were reared with normal hearing, control animals, and with the form of conductive hearing loss that I just described to you. On the x-axis is amplitude modulation rate in hertz. I'll remind you that 4 hertz is the no-go stimulus. And those rates above 4 hertz, the animal gets paid a water reward for. On the y-axis is the signal detection metric that Melissa described, D prime. So let me just remind you that 0 is chance performance and 4 is essentially perfect performance. And what you can see is that the control animals, the curve of control animals is shifted to the left, meaning that their sensitivity to amplitude modulation rate or discriminating amplitude modulation rates above 4 hertz is better than that of animals reared with hearing loss. So the orange curve is shifted to the right. So that's our starting point. And then we implant animals with the same electrodes that Melissa described and the same sort of recording characteristics that she described. And we begin to then obtain neural sensitivity of recorded neurons as animals are performing the task. So the next slide shows population data from all the neurons we've collected in control animals, 289 neurons, hearing loss animals, 280 neurons. Each come from 5 animals apiece. And what you can see is that once again, the neural sensitivity of control neurons is shifted left on this curve. That is, neurons on average are more sensitive to AM rate in control animals than those reared with hearing loss. And I'll just put up the behavioral data, the psychometric data, as dashed transparent lines to remind you where they fall. So control animals, their behavior largely overlaps the neurometric performance. The hearing loss animals, their behavior is worse than controls. Their behavior is a bit better than neural sensitivity would lead us to believe. But of course, we have to factor in perceptual learning, with which Melissa talked about. And so we expect that's one of the reasons why ultimately they do well with poor neural sensitivity. So Melissa also mentioned the effect of task engagement, which she referred to as top-down processing. And we were interested in whether animals reared with hearing loss also displayed a differential effect of being engaged in the task versus being disengaged. So we performed first the experiment I've described. We looked at the behavior of animals as we recorded from cortical neurons while they're engaged in this amplitude modulation discrimination task. And we compared neural performance for the exact same neurons as animals sat in a cage without a nose poke or a water spout and listened to the identical sounds. So we called this the disengaged case. Now, I'll put up the graph that I showed you before of neural sensitivity for animals engaged in the task. And I'll remind you that animals reared with hearing loss displayed neural sensitivity that was much poorer than that of control animals. The curve is shifted rightward. When we looked at those same neurons, both control and hearing loss, while animals were disengaged in the task, we found that in each case, neurons performed very, very poorly on the AM discrimination paradigm when we quantified that with signal detection metric. And what that says to us is that there is a smaller enhancement during task engagement for animals reared with hearing loss compared to those animals that grew up with normal hearing. And we're exploring this as a potential proxy for a cognitive deficit suffered by animals that grow up with hearing loss. So let me summarize what I think we've told you. We've used the TBSI head stage. It permits real-time assessment of cortical sensitivity in freely moving animals. And for that, that was crucial. Gerbils don't adapt very well to head-fixed recordings. On the other hand, they're easily trained and they perform wonderfully in the freely moving paradigms that we've presented to you. Melissa showed you that perceptual learning is associated with modulation of top-down signaling to auditory cortex. So we think that the plasticity actually occurs outside of auditory cortex and is conveyed to auditory cortex during task performance. And then I told you that following a period of developmental hearing loss, top-down signaling becomes weaker. So we also believe that we have evidence for modulation of top-down signaling following a developmental manipulation. And that ends our formal presentation.
Hello everyone, my name is Bradley Barth, and I'm a PhD candidate in Dr. Xiling Shen's laboratory at Duke University. And today I'll be talking to you about some of the implantable devices that we're using for neuromodulation of gastrointestinal function. In the Shen lab, we study control systems in the gastrointestinal system. Such of these projects include neuromuscular control of motility, epigenetic control of colon cancer, and feedback control in the intestinal stem cell niche. Now today we'll only be talking about this first project, the neuromuscular control of motility. So today I'll be referencing functional gastrointestinal disorders, or FGID. They're a collection of symptoms or disorders that affect 20% of the population. Now unfortunately, the diagnosis of FGID is based almost entirely on a patient's symptoms, which can be very severe and debilitating. While the causes of FGID are not known, they're thought to be associated with long-term damage to the enteric nervous system, or the nervous system that resides in the walls of the gastrointestinal system. If we look here, the enteric nervous system consists of enteric ganglia, shown in red, that physically exist in the outer layers of the GI system. So in the bottom of both of these figures, we see the villi, or the finger-like projections that extend into the center or lumen of the gut. And the outer layers include both muscle layers and enteric ganglia. Now the difficulty here to get accurate recordings is that we're not always certain whether we're recording from neurons or from muscle fibers. Furthermore, when we record from the electrical activity that contributes to motility in these systems, we need our research subjects or animals to be awake. That way that the motility can replicate what's the normal physiological conditions. So here we sought out to be able to record this electrical activity using TBSI's wireless implantable device. So as I mentioned, gut motility is dramatically affected by anesthesia. So in this case, we need our animals to be awake. Therefore, it's also best for our system to be wireless and fully implantable. That way we can record from animals in a freely behaving environment. Now, in order to test this device, we decided to record EMG activity from the hind limbs in rats. We picked the hind limbs in rats because it's easy for us to apply a very specific stimulus by pinching the hind toes of these animals and watching the leg twitch response and measuring that specific event, rather than trying to correlate any unknown electrical activity with motor activity in the gastrointestinal system that's more difficult to evoke specifically. So here we went about implanting TBSI's wireless implantable device for electrical recording in the peritoneal cavity of a Sprague Dawley rat. So that's shown here as we insert this TBSI 5-channel recording device into the peritoneal cavity and suture the abdomen closed. We then went on to place 2-channel EMG surface electrodes on the hind legs of both legs of this particular animal, so we could do real-time recording in an awake and freely moving animal. The recording configuration looks something like this, with our research subject in this cage surrounded by this white inductively powered cage collar. The inductively powered cage collar supplied power to the device without any wires or significant temperature increase in the device. We collected our signal using the TBSI 5-channel receivers with the antennas shown here, and identified by this lower LED, we've locked onto our channel in this receiver. And this is an example of what the recording configuration might look like. Now, we see our animal test subject in this physical restraint system below, so we could get a good hold on its on the toe pinch technique while recording EMG. Again, the antennas are placed outside the animal, and the animal is located in this inductively powered cage collar. Again, you can see that we have good channel lock with our wireless system, indicated by the lower LED shown here. So when we go out and pinch the toes of this animal, we see a very clear EMG response on all 4 channels. Now, when we pinch the left leg, it seems that all 4 channels respond to this twitch, likely caused by the animal moving around and twitching both legs. But when we go on and twitch the right leg, we see a very specific response, mostly on only 2 channels, which is exactly what we were looking for. Now, this was just a simple test procedure that we were doing, and unfortunately, most of our research is still unpublished, so I won't be able to share the specific results of the work, but this is mostly to illustrate the functionality of our method. So here we see a brief video of the animal freely behaving, and the electrical data is displayed on this screen shown here. As we see, our test subject is doing well and exploring the area. And when the test subject stands down and comes back up, we see a change in the electrical activity as it walks around on its hind legs. Again, this is still using the wireless and fully implantable device. We then went on to see if we could actually modulate gastrointestinal function by stimulating the sacral nerve in mice. Sacral nerve stimulation is an FDA-approved therapy for treating motility disorders such as fecal incontinence, and it's also an experimental therapy for treating things like constipation. Now, this therapy is applied clinically in awake patients, so it makes just as much sense for us to experiment with this technology by stimulating in awake mice, again using this wireless and fully implantable device. So here we'll show 3-channel stimulation with an implanted device in mice, and we'll find the motor threshold in an acute in vivo experiment using a non-survival prep. And then finally, we'll demonstrate sacral nerve stimulation in an awake animal. Here's a schematic of the way that these leads are implanted into the sacrum. What we see here is a portion of the spine in a mouse, particularly the sacrum, which has these holes through the sacral bone called foramen, and we implant the electrodes by inserting them through the foramen to stimulate the sacral nerves. What we see here is the video of the acute trial where we stimulate the sacral nerves to achieve an evoked motor response. I'll play this video, and what we should see is the tail and hind limbs, the tail and hind limbs will start to twitch during the stimulation. So there we see stimulus turn on, and the tail and hind limbs start to twitch as we rotate through the different channels of stimulation. Now, as I mentioned, this animal is anesthetized in a non-survival procedure. Once we identified an accurate motor threshold, we went on to implant the device surgically in a survival experiment. So here we see that surgical procedure. The device is implanted into the peritoneal cavity of this animal, and the electrode leads and counter electrode are tunneled under the skin towards the sacrum, shown here, where then inserted into the foramen, and all wounds are sutured closed. We finally have a brief video of this animal awake and again in this same inductively powered cage collar. And we turn on the stimulator and watch the hind legs twitch again. Here we go. So again, I apologize I wasn't able to share some of the results of our current experiments and studies, but I'm glad that I was able to share some of the methods we've used to be able to record from muscle activity, as well as elicit sacral nerve stimulation in awake, freely moving animals. Today I've shown multi-channel EMG recordings in awake, freely moving rats, as well as 3-channel nerve stimulation in awake, freely moving mice.
So we're going to start our Q&A session with a couple of questions that have come in ahead of the live presentation. Irma had asked, how many channels simultaneously can be transferred wirelessly and at what sampling rate?
We can simultaneously transfer up to 127 channels at a sampling rate of 25 kilohertz per channel. And also a 62 channel, we can go up to 50 kilohertz per channel.
Okay, perfect. Brad, this question is for you. What stimulation parameters did you use during your experiment?
So during our sacral nerve experiment, we were stimulating at 300 microsecond pulses, repeating at 10 hertz, with each channel stimulating for about 3 seconds each.
Perfect. Our next question is, how long can the device actually be left implanted without compromising signal quality? And this is coming in reference to aging studies. Taylor Vinson, would you be able to answer that question?
Yes, it will be 120 days, which will be 4 months.
Okay, perfect. And Lorenzo would like to know, can subjects being studied be housed in groups, and what is the effective range of the receiver in relation to the distance from the head stage?
We can do 3 animals simultaneously. Each one is on a different frequency, and the range of the receiver is about 1 meter.
Okay. Dan or Brad, do either of you work with this during your research? I mean, housing animals in groups, or have any input on the range that you guys receive?
For the cortical recordings, we have housed animals in groups. I think it's less an issue of the TBSI device, because we only place the TBSI head stage on animals while they're performing, and we never have animals all performing in the same arena at the same time. And I think the question about distance of the antennae is pretty much what's already been said. Okay,
perfect.
I'll add to that, if I may. The very nice thing with the implantable device is that, for me at least, is I can house animals together and not worry about them picking at their own implants or each other's implants if they're entirely implanted.
Okay. Dan, this question is for you. You guys have recorded many different configurations. How does the wireless TBSI technology compare to making these measurements in a more tethered approach?
So we've used the same electrodes with tethered recordings directly to Tucker Davis acquisition system and with the TBSI system, and the signal to noise was identical. So our sense is that signal to noise is determined by the electrode itself, not by any of the electronics that come afterwards.
And TBSI, this question is for you. Geet had asked, can you simultaneously stimulate and record neuronal activity in real time?
Yes, we can. They're on different frequencies, so they can be controlled separately and record separately.